Welcome to the official kickoff of our Innovation 250 speaker series. The National Labs for the 250th birthday decided to talk about innovation. And here at the lab, it's part of who we are, it's part of what we do, one of our core values for all we do at the lab. Across the national landscape, people are trying to understand better what leads to success. In other words, what does it actually take to move from scientific discovery to a technology that could really have impact for society. We're taking the opportunity to have a conversation with a few people on that topic. To get us started, we're joined today by someone who's not only redefined the boundaries of chemistry and biology, but has also fundamentally changed aspects of how we treat health and disease, and has started some companies around that. Carolyn Bertozzi is a pioneer of bioorthogonal chemistry. After completing a postdoc at UCSF, she came here, and of course, famously, as well as the College of Chemistry, she was famously director of the Molecular Foundry during its formative days. And she's currently a professor at Stanford, director of an institute, and a long list of titles around that. And her numerous words, I only bring up the 2022 Nobel Prize in Chemistry because I don't have half an hour to go through all the others. But I want to say, among all the great scientists I have known, some become leaders of people building institutions. Others of them have become scientists, entrepreneurs, building companies. Very few actually have done both. And she's one of those people who has done both. So please join me in giving a warm welcome to Carolyn Bertozzi.
Thank you, Mike. It's such a great invitation to participate in this really great series. I think it's a wonderful way to celebrate the nation's birthday. It's always fun to come back to Berkeley and to drive up the hill up to LBNL. And there's always a new building that causes me to be disoriented and couldn't find the cafeteria, you know. So, but this room hasn't changed a bit, I could say that. So thanks everybody for coming out to spend your lunch hour, celebrating science here at LBNL. And Mike, you gave a really nice introduction. I thought I would start with, this is kind of like the tagline of my lab as it currently exists. I'm the director of an institute at Stanford, which goes by the name of Sarafan, my laser pointer, there we go, Sarafan Chem-H. So Sarafan Chem-H, the Chem-H part is an acronym, and that stands for chemistry, engineering, and medicine. So the chem is an acronym, and the H is for human health. Okay, so as that unwieldy acronym might suggest, this is an institute where we brought together people from lots of different scientific and engineering backgrounds with the common purpose of trying to improve human health for the future, for posterity. And knowing, as you do, how interdisciplinary and how forward-thinking LBNL is and has always been, it won't surprise you that I was very much inspired by my learnings up here at this institute over a span of maybe 20 years before I moved to Stanford and deployed those learnings in this new context of human health. But the work that I thought I would tell you about, which was kind of the early work that kickstarted my lab at Berkeley, it was all done down in Latimer Hall. With Berkeley graduate students, which kind of set the tone for a career, as Mike suggested, which has been marked by dual interests, both in organizing and coordinating people in the academic center towards educating the next generation and producing knowledge and innovations, to spinning out technologies into the commercial sector and trying to make products that benefit human health. And so I'll show you some examples of science that have taken me in those directions. First and foremost, so it's on Zoom, right? There we go. Just a little background about myself and how I came to be. I'm from Massachusetts. I was born in Lexington, which is an historic town. And when we say it's the 250th anniversary, it's the 250th anniversary of the battle that was fought right there, okay? That's the Lexington Green. And what happened was, the British arrived and stormed the coast of Massachusetts, and the British soldiers marched and fought their first battle against the revolutionaries, the colonists, right in that location. And that guy, the statue, he's the Minuteman, right? So that was the name of the soldiers of the revolution that kind of fought back in their scrappy little way. And on April 19th in Massachusetts, we celebrate Patriots' Day. That's a holiday in the state. And we're celebrating this moment of the revolution, of the birth of our independent nation. And there's a parade in downtown Lexington that ends right there in the Lexington Green, as it's called, where everybody's dad puts on a uniform and reenacts the Battle of the Revolution. So when I grew up, I was like, oh, that's my dad, and if your dad was like a British soldier, it's like, boo, And after this fake battle with muskets and everything, and Paul Revere rides on a horse and says, the British are coming. I mean, the whole thing is quite dramatic. Then everybody goes and has pancakes and sausages. And then by then it's 7 a.m. All this goes between 5 a.m. and 7 a.m. And all the kids go home and take a nap after that. So that's my memory of the birthday of our nation. The reason I was in Lexington is because my dad was a professor at MIT. And a lot of professors lived in this town and would commute to Cambridge. And so I grew up in a household that was a physics household. He was a nuclear physicist, so it was a Department of Energy household. And even though I wasn't instructed that I had to become a scientist, that really wasn't part of the deal, I will say that I was protected as a young girl interested in science. And I was born in 1966. Those were times when girls and women were not encouraged to pursue careers in science or careers at all, for the most part. But in contrast, my dad, kind of created a science bubble around his three daughters so that by the time we were old enough to be infiltrated with these negative messages, it was too late. We already thought we could be scientists. And so 2 out of the 3 of us are scientists. I went to Harvard as an undergrad, which was a profound insult to my dad. Not because it wasn't MIT, but it was because I wouldn't get the free tuition perk of MIT. It was really, really the point. And he said, wow, you're half a mile away from a free university education, and I'll tell you what, here's the deal. If you change your mind and go to MIT with the money I'll save, I'll buy you a house. I don't know that that's still true, although I think housing and college prices have maybe escalated at roughly the same trajectory. But at the time, I really didn't know what I wanted to do and major in. And I thought I might want to do something outside of science, which would make MIT not the ideal university. As it was, though, I discovered the field of chemistry when I was in college. And by the time I emerged from Harvard, I was pretty dedicated to organic chemistry. And that's what I wanted to pursue with a PhD. So, grew up in Lexington, went to college 20 minutes from home. In my senior year, I was walking to class one day with my backpack, and I crossed Mass Ave, if you know the geography there, and a car drives by and honks, and it's my dad on his way to work at MIT. I'm like, hi, Dad. So that was the moment I kind of rolled out the map. I'm like, all right, where can I go that's as far away as one could fathom at the time? And so that's what took me to fabulous UC Berkeley. So I arrived in Berkeley as a first-year grad student in chemistry in 1988, and I joined the lab of a brand new professor, and his name was Mark Bednarski. And Mark had just finished his postdoc at Harvard, so I had met him at Harvard, and he had worked in George Whiteside's lab. And many of you will know George Whitesides, who's a legend in all areas of science, but especially in chemistry. And so Mark had all these cool ideas about doing synthetic chemistry on sugar molecules to make biological tools that could be used to study glycobiology. So he was the person who introduced me to glycoscience, which is an interest that has stayed with me till this very moment. And I did my PhD in lovely Latimer Hall. It was actually lovelier than this at the time because it didn't have these big earthquake-resistant crossbeams. So the balconies were more open and you had a better view. And then some years later, they came and said, well, we don't want the building to fall down in an earthquake, so we're just going to staple 3 feet of concrete to the outside. And so that's what happened. It's okay. Hathcock Hall looks gorgeous, by the way. I have to say, that new building, it's glass and beautiful. And so that's going to be a really fun place to visit. But, in the course of doing my PhD in Latimer Hall, I started reading more and more about the biology of sugars, though I had focused on the synthesis as a grad student. And to delve more into biology, I decided it was time to jump ship and go to a completely different non-chemistry environment. And at the time, that was UCSF, the medical center, which was exclusively at the Parnassus campus. This was before the Mission Bay campus. And I worked for an immunologist named Steve Rosen. And he's the person who first introduced me to the role that cell surface sugars play in mediating cell-cell communication. So for those outside the field of biology, maybe I'll be the one to tell you that all of your cells are coated in complex carbohydrates. Your cells are balls of sugar, essentially. And those complex carbohydrates have lots of interesting structures. They're very structurally diverse. And some of those structures are there so that they can bind to receptors on another cell. And this is one of the important roles that cell surface sugars play in multicellular biology. They allow cells to touch and communicate and share information. And I found this to be really fascinating. And so my first academic position was back at UC Berkeley after my postdoc. And it was focused on developing chemical tools, which I had become, pretty experienced in for my PhD, and applying those tools to study the cell biology of these cell surface carbohydrates, which is something I had learned in my postdoc. Now, at the time, in the world of glycobiology, it had become clear that the structures and the patterns of those sugars on cells were not random at all. They were very specific to cell types, and they changed when cells went from being healthy to being diseased. And in cancer in particular, there's some very well-known, quite dramatic changes in the architecture of the cell surface sugars. Just as a case in point, one of those building blocks in your cell surface carbohydrates is this monosaccharide called sialic acid. And all of your cells have glycans or complex carbohydrates that terminate or are punctuated with a sialic acid. That's normal, but usually they're kind of a well-manicured garden, kind of sparsely distributed, very neat and tidy. And in cancers, things change where that neat, tidy garden becomes overgrown into a tropical jungle, which we would call a hypersialylated glycome, a very common feature of cancer. And this intrigued me back in the early 90s, late 1980s, because it seemed to me that if there's a molecular change like this, there's an opportunity to exploit that molecular change for diagnostics or maybe even to target therapeutics. And so when I started my professorship at Berkeley, I came back to the chemistry department. This is now 1996. The goal of my fledgling lab was to develop a strategy for molecular imaging of cell surface sugars so that you could see these kinds of phenotypes emerge, ideally non-invasively in the human body. That was the vision. And the 1990s was a really great time to start thinking about molecular imaging because there had been investments in new tools, and some of these tools were really radically changing the way that we visualized biomolecules in living systems. Case in point, the 1990s was the decade where the green fluorescent protein was discovered and where Nobel laureate Roger Chen, a former Berkeley faculty member, had taught the world how to use this as a genetically encoded fluorescent probe. So you fuse your protein to the GFP, and then you could see that fusion protein in live cells using fluorescence microscopy, even in live animals like fish and worms and transgenic mice. And in the world of nucleic acids, some really cool new tools had been developed to image transcripts or genomic sequences using base pairing, hybridization of fluorescently tagged oligonucleotides. And I was really jealous because the protein folks could image their proteins, the nucleic acid folks could image their nucleic acids, but what about the glyco people? We couldn't see the sugars on our cells. There was no imaging platform for the sugars. So the first little group of students to join my lab at Berkeley sat down and thought, all right, let's find a way to do this. And we were inspired from some biochemistry work from the lab of Werner Reuter, who was a German biochemist who studied the enzymes involved in the biosynthesis of sialic acid, that sugar that's overgrown on the cancers. And he had published a paper showing that you can feed cells a modified precursor of sialic acid. It's a sugar that would go by the name N-acylminosamine. And you could put small functionalities in this R position that were unnatural. And as long as their size was limited, that metabolic precursor would be processed by all of the enzymes in the biosynthesis of sialic acid, leading ultimately to the attachment of that modified sialic acid onto the glycans that populate the surface of the cell. So you could literally use the biosynthetic pathway as a vehicle to deliver some modified side chain into a cell surface sialic acid. And that became the inspiration for our first cell surface carbohydrate imaging platform. It was a two-step process that we developed, where in the first step, we would chemically modify a simple sugar with a chemical group that was reactive. We would call it a chemical handle. And we would feed it to cells and let the cells do all of the biosynthetic work and integrate it into a cell surface glycan. And the key here is this X group, this chemical handle, has to come along for the ride. No side reactivity with any biological functional groups. It has to be inert in this setting. But it has to have a special reactivity with another chemical group that we would attach to an imaging probe, like a fluorescent dye. So that in the second step, when you add the probe molecule into the biological system, the X and the Y react with each other, form a bond, and now you can see the sugar. But the problem is you need a chemical reaction between an X and a Y, where they'll form this bond in a biological system without any interference. And that was an unknown chemistry at the time. So the need for that chemistry, the need to identify the X and the Y, that's what led to the development of what we later called bioorthogonal chemistry. So literally chemistry that is orthogonal, so not interacting with biology. And to make a long story short, a few years into this process, we developed the first such reaction, which we called the Staudinger ligation. It's a reaction between azides and phosphines that leads to the formation of an amide conjugated product. And we got some mileage out of this chemistry, but there were properties of the reaction that made it not ideal for imaging in vivo, not the least of which is that it's kind of a slow reaction at physiological temperatures and just took too long. A couple years after that, Morten Meldal and Barry Sharpless, with whom I shared the Nobel Prize in 2022, developed another reaction of the azide in which they conjugate it to an alkyne catalyzed by copper. This is the so-called copper-catalyzed azide alkyne cycloaddition, much more popularly known as click chemistry. How many people here have done a click chemistry reaction? Probably there's somebody here. Yeah, there's always a few. It's nothing to be ashamed of. It just isn't fine. And that's a great chemistry too, but not so great for biological imaging because the copper catalyst is toxic. And so we had a third reaction from our lab in which we eliminated the need for copper by putting that alkyne into a strained ring system, which enhanced its reactivity. And that we call the copper-free click chemistry reaction. And these three reactions, which all start with an azide, have become really popular in biology and in pharmacology and medicinal chemistry and even material science. And we use this process to image sugars. We would take simple sugars, for example, this derivative with an azide, feed them to cells. They get converted to another sugar, which is in a cell surface glycan. And then we use the copper-free click chemistry to click on the probe and visualize those sugars. And here is a picture of a zebrafish that got labeled several times with several sugars, different colors. And we spent some years mapping changes in cell surface sugars during embryonic development and during the development of tumors. But for this particular chat, which we're going to have in a minute, since the theme is innovation, I thought I would just spend a few slides showing you how some of these chemistries have left the academic environment and been deployed in the biopharmaceutical industry to make new kinds of medicines. As Mike mentioned, I've spun out companies from my lab, and here's a bunch of them that are shown on this slide, where each of these companies is a company I co-founded and which relates to a new kind of therapeutic molecule that is a hybrid of multiple proteins or proteins and small molecules or polymers and sugars. And anytime you need to link together large, complicated molecules like proteins and sugars and synthetic drugs and so on, bioorthogonal chemistry is probably going to be a useful tool because you can take a big, complicated molecule like a sugar on a cell and still do a very selective chemical reaction on it without any interference. And that has use in a lot of different environments. For example, for making antibody drug conjugates. And if you haven't heard of them, let me be the one to tell you that this is a very exciting new class of drugs, new meaning, 10 or 15 years old in terms of being used by oncologists, that basically targets very toxic chemotherapy drugs just to the cancer cells and keeps them away from healthy cells, which makes the drug much less dangerous and unpleasant for the cancer patient. So an antibody drug conjugate, basically you stitch the drug, a synthetic molecule usually, onto an antibody where the antibody binds a tumor antigen. And so the antibody directs the drug to that tumor cell, where after antigen binding, it'll get internalized by the cell. The cell will deconstruct it inside, and that releases that toxic drug so it can kill that cell without too much bystander killing. It sounds simple, but the details are difficult, and it's taken decades for people to optimize these molecules and make them as effective as they are. People had to work out, how do you make good antibodies? People had to work out what kinds of drugs might work in this setting. People had to work out, how do you link the drug to the antibody so that the linker is stable in circulation, but will fall apart inside the cell? And all of these problems have been iteratively solved over some decades. But the problem that we took on had to do with the attachment site. How do you actually make that bond, the literal chemistry biology interface, right, between the protein, which you made in cell culture, and the linker and the drug, which you made chemically in the laboratory? And once again, bioorthogonal chemistry is really useful for this problem because proteins are very highly functionalized with all the 20 amino acids and many reactive side chains like lysines and cysteines. If you can introduce a new chemistry into a protein, a bioorthogonal functional group, then you can make sure that when you attach a linker and a drug to that antibody, it goes exactly where you want it in a homogeneous way with a controlled ratio of the drug to the antibody. And these are features of modern antibody drug conjugates that were difficult to achieve with old school reactions that weren't bioorthogonal. I even started a company from Berkeley, from chemistry department, with one of my former PhD students to translate one of our methods into the commercial setting. And that company was called Redwood Bioscience, and it was acquired by another company called Catalent back in 2014. And basically, this is a technology where we use a naturally occurring enzyme, the formyl glycine generating enzyme, to oxidize a cysteine side chain within a 5-residue consensus sequence to the corresponding aldehyde, the formyl glycine residue. So any antibody that we express in cells that have this FGE, that antibody, if we basically genetically introduce this sequence, will come out of the cell with aldehyde groups at the site genetically encoding that cysteine. And then we invented a chemistry that's selective for aldehydes that you can do on a protein, and we know exactly where the chemistry takes place and exactly how many drugs are attached. And we're not the only company that has leveraged bioorthogonal principles for site-specific antibody drug conjugates, as we call them. There's several others here, and 2 of these are actually in the Bay Area, those being Sutro and Ridge Bio. So I think it's a really great time to be thinking about translating technologies from the chemistry-biology interface into the therapeutic world, and hopefully you'll be seeing more of that. And with that, happy to join the fireside chat.
Very good.
Thank you.
So thank you. That was great. And also hearing about Lexington. So I want to follow up on some of the things you're talking about. You obviously have a very broad research portfolio, and some of that research led to innovative technologies and companies. So how do you, when you're considering your research directions, do you think at the beginning, oh, this is one that could lead to a company at the end? Or do you say, you do your research and somewhere along the way say, this is pretty cool, this could be a company. How does that work?
Yes, all of the above. So, the funny thing is in the early days of my independent career, I never thought about companies.
Yeah.
It was all basic science, trying to create new technologies that could be useful for scientific research. To the extent that I might have articulated applications down the line, that was very secondary. I was much more focused on, curiosity. Right?
Yeah.
Can we invent reactions that you can do on cells? The answer could have been no. Right? We didn't know. But then the older I get, right, the more I am motivated by thinking about the practical use of what we're doing in the world outside the lab. That started kind of later in my 20 years at Berkeley. And what happened was I had the student who formed Redwood Bioscience with me, he wanted to start a company out of his PhD, but he didn't know what that company would relate to. The work he was doing in my lab was not related to this project. He had a completely different project that was not really a translatable project. But he wanted to be an entrepreneur. And so he came to me one day and he said, are there any projects in the lab that you could imagine are kind of ready for translation, and which ones would they be? And I said, well, this aldehyde project, we could use this for therapeutic bioconjugates, And so he said, okay, I'm going to spend the last 6 months in my PhD learning that project, which was not his project. And I'm going to spin it out into a company with you. So that was the first time that we kind of thought like that. And then by the time 2015 rolled around, I had become more and more interested, because the older you get, right, certainly in the world of drug development, there's a long timeline on it. So if you have an idea today that you think could lead to a drug, if everything works really well and perfectly and everything, maybe 15 or 20 years from now, you have the drug, okay? So that means the older you get, you have to think about every new thing you start, you need a 10-year runway. And you start running out of 10-year runways as you get older. And so, at some point when I was kind of in my late 40s, I started to think, if I'm really going to try to make medicines from these discoveries, I'd better get going on it. And unfortunately, what that led to was me leaving Berkeley and going to Stanford, because Stanford has a medical school and medical center, and it's just easier to do translational medical research at Stanford. Although now I realize it's much harder to do translational other types of research like energy science at Stanford. There you'd want to be here.
Okay.
Yeah.
Okay. So you talked about this, going from being, in some sense, a pure chemist to being an entrepreneur chemist. So once you get into that, do scientists, innovators think about their research differently than scientists who are never going to do that?
I think I do. I'm nowadays at Stanford, I'm much more inclined to start a new project with pitch a thesis project to a new PhD student that I think might have translational potential. Which isn't to say, if a student came to me and said, I'm really curious about how this thing works, I would really like to get into it and try and figure it out. I'm on board with that. That's great. But honestly, if it's coming from me, I'm thinking more about, it's really hard to get drugs into the brain. Let's work on trying to get drugs into the brain. I'm more likely to pitch a project like that.
So another aspect what you talk about, you work in science, engineering, medicine altogether. So very different fields. And so a lot of people in university know of C.P. Snow had a famous lecture on the two cultures and the scientific revolution. And he talked about the two cultures of science and humanities and the gulf of mutual incomprehension between them. So it was just great, which I always remember. But we in science know, as we look around the laboratory, science is not one culture, it's 14 or 18 who actually, in turn, don't talk to each other very easily and use different language and different ways to work. And our laboratory is a celebration of that. So how do you think about that? You've been in a situation of bringing a collaboration together from these different parts with different languages. So how do you see that? Is that a barrier? Is that an opportunity? Maybe some of both? How do you think about that?
It's a huge opportunity. It really is a huge opportunity. I first recognized that when I went from Berkeley PhD student to UCSF postdoctoral fellow, because I had left the world of chemistry and entered the world of immunology. And who's a PhD student? Anybody here? No. Okay. So you live in a little world, right? And in every of these little cultures, little subcultures, there's a way of doing things. There are things that are considered exciting and hot. Oh, this is hot. There are people that are the lore, the sort of the grandfathers and mothers of the field who important people in your field. And you think, at least I thought, that was like the universe, right? And then I went over to UCSF, and on day one, I was disabused of any notion of these things because nobody in my new lab had heard of anyone in my former world. People I thought were so important, really important people, and they'd never even heard of them. The journals I had published in, they had never heard of. And all of a sudden I was like, wow, all of it was artificial. There's nothing universal about it. And likewise, they had their own important people that I had never heard of. They had their own important journals. And so that was a bit of a revelation. And I found that liberating because sometimes when you're in these bubbles, it can be suffocating, because if you're not one of those important people working in those important things, you feel unimportant. But the truth is that there's a much bigger world than that. So that was great for me to kind of learn that and understand that. The other thing I realized is they had all of these frustrations and problems that they assumed, because that was their groupthink, were impossible. And I came in from the outside as a chemist, and I was like, well, let's just invent that thing, right? And I'm talking simple things like the green fluorescent protein. Those of you who've worked with it might know it matures. It doesn't just come right off the ribosome fluorescent. It has to mature into this folded protein, and there's an oxidation chemistry that happens to create the fluorophore. So you cannot use GFP as a marker in anaerobic environments because it won't mature, right? It needs oxygen. And people are like, yeah, it's too bad we can't use GFP to study, for example, the gut microbiome, which is an anaerobic environment. And I'm like, well, why can't we invent a fluorescent reporter that's for anaerobic settings? And they're like, you could do that? Right? and as a chemist, you love problems like this. Right? Like, oh yeah, let's think about that. So that was also very interesting. And then after I was on the faculty here, through a weird set of circumstances I won't get into, I had the opportunity to launch a program in the material sciences division. Now I wasn't a material scientist. Nobody would have looked at me and said, yeah, she's a material scientist. Not at all, okay? But at the time, there were people with visions that realized that biomaterials could be important, and maybe we want that represented at LBNL. So I joined material sciences, and I got a grant, and set up a little pilot lab right over there in that building. Whatever the number was, I forgot. 66, Chef Neaton. So yes, and then all of a sudden I'm up there hanging out with people who are making carbon nanotubes. Right, and I'm like, carbon nanotubes, what does that have to do with carbohydrates and cells and stuff? And that's when all these interesting ideas came up. Right? Oh, we want to use carbon nanotubes for drug delivery applications or as biosensors, but carbon nanotubes they don't dissolve in anything. They're charcoal, right? They're a chunk at the bottom of your tube, and how can you manipulate them? And so there I was, and I said, well, what do cells do, right? Cells have a hydrophobic membrane, and yet they're happy in water. Why is that? Because they are coated with sugars, which are hydrophilic. So I'm like, well, let's coat the carbon nanotubes with sugars. And lo and behold, boom, they dissolve in water. And we did this with carbon nanotubes. We did it with boron nitride nanotubes, and all kinds of fun. And I made friends with all these other scientists, physicists, and people who are actually real material scientists. And I learned a lot from that, like Young's modulus, I know what that means. And, stuff like that. And that was enriching. I also met Arun Majumdar. Okay. And he taught me about the incredible science of building efficiency. And, I've been in buildings before, but it never occurred to me that someone designed this thing, and there might have been a lot of science behind that, and there is, and that was fascinating. And now Arun is the dean of our Dorr School of Sustainability at Stanford. And he and I are launching a new initiative that is the interface of human health and planetary health. And none of this would have happened if I hadn't had the opportunity to spend time at LBNL and meet scientists completely outside of my own space. So that's my lesson in life.
So I try to pivot to something else. Talk about as you're in your role as mentoring a lot of early career scientists, and talk about how they choose research and how they develop research, and the fact that not all research directions end up in success.
No,
I've been told.
Most of them don't.
How do you talk? And so you've got this trade-off. Do I do something safe? Do I do something? So how do you talk to your students as you're bringing them into that about how you choose that?
You're absolutely right. I just gave a commencement speech at Rockefeller University, and the theme, and I was talking to the parents in the audience, because unless you have been through the PhD process yourself, which most of their parents had not, it's hard to get your head around, what do you do? How do you get
Why does it take so long?
Or it's even worse than that, because it's like, how long will it take? Nobody knows. Right? And how different that is from an MD or a JD, right? Where you go in on day one, there's a workflow and a script and a formula, and you follow it, and you'll get your MD.
And you time out, and you're done.
You're 4 years. You're done. If you do this, and this, they hand you an MD. PhD, y'all know. You come in, it's like, all right, you know, what project you want to work on? Okay, good luck with that. And, you know, touch base with me every now and then and let me know what you think you should do next, you know? Right? And you're right. Often the intentions don't play out in the real world. Sometimes the person stumbles into something exciting inadvertently, and that causes their research to pivot in a really exciting new direction. More often than not, they hit their head against the wall and they can't figure out how to break through. So it's incumbent on me to really be able to read the room with them and understand, first of all, like, are they making progress even though they might not feel like they are? But do I see that there is an enhancement of understanding, of learning, and that's valuable. Will they, you know, there's a pragmatic issue here. They have to graduate eventually.
Yeah.
And we don't want it to take more than 6 years, certainly. And they need to have a story to tell so that they can get a job, right? They have to give job talks and stuff. So if I see that they're creeping up into their 4th year and they still don't have what I would think of as a compelling job talk, like a story to tell that they own, then I'm like, all right, I need to intervene a bit here and figure out how can I get them onto something that will produce a story for them in a reasonable time frame. And that's on me to figure that out.
So let me talk a little bit more about sort of talking to early career scientists. And we talked a little bit about this. The pivot point in my career was when I didn't get tenure at Princeton. So that was the most important. I was talking about my career at one point. I said, yeah. And that's when I realized when I went to Santa Barbara that I needed to depend on my own self to figure out what were the right research directions for me and not to listen to these senior people who were telling me what to do. Okay, so and then everything sort of got better. So what is the one piece? So that's what I wish I would have told myself back when I was at Princeton, for example.
Yeah.
So what would you have told yourself when you're early on in that career?
I would say that my advice is don't take other people's advice. Yeah, I had some really bad advice at Berkeley as a new assistant professor. I had ears full of bad advice.
Yeah. I realize a lot of senior PIs here may not be liking what we're saying here right now.
And we're 2 of them. Yeah.
This may come back to us.
Well, because the truth is that the advice for their generation might not work for you in your generation. Right. Things change, right? And the advice I got from my senior colleagues here at Berkeley in chemistry in those first few years that I was on the faculty was absolutely not good advice for me. It didn't work for me. And I was really unhappy. Like the first 2 years, sorry, anyone, but the first 2 years I was here, I was like, every few weeks I would take the San Francisco, so this is before the internet, okay? So the San Francisco Chronicle had, at the back page was the jobs page. Remember that?
Yeah.
So I would flip to the back of the Chronicle and circle the jobs. And I was like, I could do that.
I could drive a truck.
This job I hate it. And I don't like this. Yeah. I didn't like my job and I didn't think I was good at it. And I was like, what am I doing here? And I think the fundamental problem was my approach to my job followed the script that other people had been kind of handing me, and it wasn't working for me. And so at one point I had some kind of an epiphany. I talked to my dad, who was still on the faculty at MIT at the time. And I said, Dad, I don't know what to do. I hate my job. Yeah. What should I do? And he said, well, all right. How long has it been? Well, I've been here like almost a year. And he's like, oh. He said, every job is terrible for at least 2 years. Any new job, you're going to maybe hate it for like 2 years. Now, after 2 years, maybe it's a really shitty job and you just should quit. But if it's not yet 2 years, just give it time and try and find your way there. Don't just bail out yet. All right. And he was right about that, because it was about the 2-year mark that I sort of had this adjustment in my brain where I said, you know what, I'm just going to ignore people. I'm just going to do what I think is interesting and exciting that is going to work for me and my students and have fun with my students. And then, if I can't keep my job, if they don't tenure me, life will go on. I'm sure I'll be fine and not worry about that, Okay. And that turned out to be the right approach.
Okay. So I'll do one more topic because it comes up with people, how we define success in our fields, especially in universities. And many of us are trying to change those metrics from things more than just citation indices.
Yeah.
And to make it more about impact, and innovative technology is one example. But, there's a lot of faculty review committees tend to draw back to those things. So how do you think we're doing at that overall?
Not great. Yeah, I agree that these quantitative metrics are magnetic. They attract you because they're easy.
Yeah.
It's easy to look at numbers and rank order people based on numbers. But they don't tell the story often,
Right.
about the real value of the person or the science.
Yeah.
So, I agree that ultimately, like, you have to have a North Star.
Mhm.
So for academia, right? So in the university, what's our mission, right? Our mission is to create knowledge, invent things, and teach the next generation, right? That's kind of it,
Yeah.
right? And so I think if you reward people based on, like, did they create knowledge or invent things, and have they, train the next generation? Like, if you start with that,
Yeah.
you're on the right track. And then, what's the value of the knowledge? That's hard, because the reality is most of us don't know the impact of our own science in our lifetime.
And it often takes many years for
Yeah.
that to be clear.
Yeah. And we might not ever
Yeah.
fully understand our own
Yeah.
impact,
Yeah.
because you're right, it might unfold on a much longer time scale than metric rewards have to occur.
Yeah.
So I don't know what to do about that, but
Yeah.
it's in my face all the time, because I think every day about how bioorthogonal chemistry wouldn't exist were it not for some foundational chemistry from Germany circa 1920, 1930, right? It was the work that was going on in Germany at that time that formed, like, the inspiration for these reactions I mentioned. And there's no way that those chemists in 1920 could say, oh, this is going to be great, because 100 years from now, someone's going to image glycans in a zebrafish, right?
Yeah.
So you kind of have to be realistic about this, right? So it's hard. I'm not sure that I know exactly how you reward a person based
Right.
on their science, but I think looking at their students, I think the quality of the students they're producing, the way that the students embrace science, the creativity of the students, the confidence of the students, the impact that in a way is the better metric.
Right. Yes.
Right?
Yes.
But how you do a tenure case on that, I don't know.
Yeah. Okay. Good. So let's see. We want to take a couple of questions from our audience, and so yeah.
I've got the
You're ready?
represented. I'm representing.
You're ready for that? Okay.
Yeah, I'm representing Zoom and chat here.
Okay, good.
All right. So the first question comes in, and Ian wants to know, apparently he's been reading your bio, that you're interested in music.
Yeah.
I would like to know how music influences your scientific work.
Okay. Yeah.
And the band? You gonna talk
Yeah, I
about
have
the
a band. Yeah. All right. Okay. So, music. Like many of us, my mom made us take piano lessons, when we were kids. Who had to take piano lessons? It's a universal thing. And I didn't like that much. I didn't practice enough, and my mom nagged me a lot, and it was always a point of friction in the house. And then thankfully, my mom won that battle until high school. And then I discovered jazz, and I started playing in jazz bands. And then I didn't need my mom's motivation anymore. I had my own internal motivation after that. And then in college, I played keyboard, and I joined a band that a very famous musician, not at the time yet famous, but this was Tom Morello, was 2 years ahead of me at Harvard. And he recruited me to play in his band, and that was a really great band. And he went to Los Angeles after graduation and founded Rage Against the Machine, and then Audio Slave. And now he's playing with the E Street Band with Bruce Springsteen's tour.
Absolutely.
Right?
Yeah.
What they call the Freedom America Tour, or whatever it's called. And anyway, so that was a transformative experience for me, because it was the closest I had been to somebody who ate, slept, and breathed something. And it was music for Tom. So Tom, he was like a political science major, which won't surprise people who know him. He always had a guitar in his hand, like in every lecture that he was in. And like his hand was just going in the background. Like he wasn't even paying attention to it. He was listening to the lecture, but his hands were always going. And that thing was glued to him. And I just remember thinking, like, wow, like I've never seen anybody that like monolithic about a thing, like that. And that was really inspiring, actually,
Yeah.
even though it was weird. Because then when I went to grad school, I realized that organic chemistry was going to be my guitar, right? I was going to put my whole heart and soul into that. And that was very rewarding for me. And then I kind of fell out of music because you're busy. And, I had a little apartment where I couldn't have like an instrument really. It was too loud. And this is, again, before digital technology with headsets and everything. And so I fell out of it for a long time. And that was kind of sad because I think it was really good for me to have that sort of diversity of activities, or science and then music. Music is creative. Music teaches you performing skills, which helps you deal with nerves in front of audiences. Music, if you do perform, then you have to read a room. You have to make eye contact with people to be effective as a performer. And so that's a skill that I think was useful. But then what happened was I always had how like I played the keyboard, but my air instrument was the bass. Like for some of you, it's the guitar. It's a very popular air instrument. But for me, it was the bass, and I always loved the bass. And so what happened was I'm the editor-in-chief of an ACS journal. And at our 10-year anniversary for the journal, as a gift to me, they gave me a bass, which has the ACS Central Science logo on it, a custom decorated Fender P bass. I'm like, oh my God, wow. They're like, well, you always said you love the bass. Here's a bass. And I'm like, but I don't know how to play the bass. But now I had a bass, right? I'm like, what am I going to do? And then what happened was I have a friend group in Berkeley where every January 1, we go to Saul's Diner in North Berkeley and have a New Year's brunch.
Oh, yeah.
And Corey Ralston was there, who runs the bio facility at the Molecular Foundry, okay? And she had formed a band with some guitar friends of hers. And she said, yeah, we have guitars and we do this, but, we don't have a bass. And I'm like, I'll be the bass player. And then as I was driving back from that brunch, I was like, oh my God, I have to learn to play the bass now. And so I got on YouTube and I learned to play the bass.
Oh, cool.
And so now we have a band. And we had a gig in the Kensington Circus pub just like last month. And we have another gig on July 4th in Hayward up in the hills. August 16th in Nevada or San Rafael, right? There's a little dive bar type of place. And the name of our band is Almost Pretty. And there's another LBNL scientist in our band. Do you guys, they know Eric? I don't know if the people. Hi, Eric. Anyways, so it's a bit of an LBNL Berkeley band, I guess you could say.
Cool. All right.
Yep.
Boy, I bet you weren't expecting that, were you? So we have anything more?
We do have a suggestion that we bring you back for open mic here at the lab.
Yeah, that's right. You can perform.
Yeah, right. Well, we will play any gig, and we don't need to be paid, nor would anyone pay us. So I think that works out.
Yeah.
Yet.
Food and drinks are welcome.
Right.
Maybe we'll do one more, I think, maybe, yeah.
Question about, do you think AI is, thank you.
Yeah.
Do you think AI is going to help speed up your development cycle?
Yeah, and let me say, we couldn't do a whole session like this without ever bringing up AI.
Yeah.
God forbid.
Well, we spend 90% of our time, so okay,
Right.
let's do the obligatory AI question here.
So there's that one, and also then, is AI going to impact the creative spark of research? How do you think that it will impact creative thinking and innovation?
Yeah, this is a long
That's a good one to end on.
Okay, I'll brief. I'm at Stanford. We are in the heart of Silicon Valley, right? So there's a lot of people interested in the leading edge of AI algorithm development and ways that AI can be deployed in different sectors. There's a lot of hype, to be sure. All of us are inundated every day with AI we didn't ask for, and it's annoying at some level. But the one area where I do think the impact is in the here and now is in the biopharmaceutical area, okay? And since that's an area that I engage with, I see firsthand how powerful some of these algorithms can be. So, for example, in areas of molecular science where we do have large, well-curated data sets that are formatted and standardized, legacy data, the PDB you know, which the ALF
Yeah.
here has populated nicely over many decades. That was the substance for the training of AlphaFold, which I think nobody would dispute has been transformative, certainly for my area of research. So people who work with large genomic data sets, which are now abundant from, you know, thousands of species, have used AI to discover, you know, new genes, organizations of genes. That has been transformative, no doubt. In my institute, we are launching an AI initiative, as one does, and it's focused on what kind of data do we need to train the algorithms that we really need for tomorrow. Because once you go past the PDB and genomic data sets, it's a bit of a desert, especially for molecular science. So I'm of the mind that one way that we in my institute can contribute to this revolution is to create an infrastructure for data generation. And the kind of data that we are uniquely equipped to generate are data around the functions of proteins, molecular functions of proteins. So, you know, the PDB is the photograph that trained AlphaFold. We want now the video. And the video is function, motion, catalysis, things like that. And so, you know, we've set up a robotic center for high throughput data generation around like physical constants of protein function. So for an enzyme, substrate binding kinetics, you know, KMs, KCATs. For transcription factors, you know, DNA binding kinetics and energies and selectivities and so on. And when I say high throughput, I mean like hundreds of millions of data points per week. Hundreds of millions, because that's the scale that you need to generate the trillions of data that you would need to train algorithms for something as complicated as protein function. So we are trying to play in the space from the standpoint of data generation.
Yeah, and that echoes much of the discussion here at the laboratory, as you might say. As the thing that I say in broader groups most often is the limit of AI and science is usually not the size of the model, but the quality and size and character of the data set.
Exactly.
And that was, as we've talked about many times, Protein Data Bank, of course, was the thing that led to the sudden discovery through AlphaFold, and that had been collected over decades of hard work.
50 years, right.
Yeah. And not many of our fields of science have that those data sets without a lot of work. And that's actually what's coming up all over the laboratory is that data work that has to be done so that we can actually make use of the AI.
Yeah,
that's
absolutely.
coming up everywhere here.
Right.
Yeah.
And I'm anxious about the fact that infrastructure for climate data, ocean data, has been diminished. Yeah. Because those were areas in which we had a lot of data that were actually really useful for training models. Right. So why sacrifice that?
Yeah. No. Okay. So anyway, I think we'll probably close here. But I want to come back and say first of all, what an incredible set of accomplishments you've had. Thank you for spending time with us. And we're so proud to have been part of that story.
Huge part.
And so with that, we'd like to thank Carolyn.
Thank you.